I will try to start with some preliminary consideration, followed by also a description of the functional anatomy of the basal ganglia, its neurochemistry, and then a look at striatal neuronal subtypes and the role of plasticity in this phenomena, and then trying to, of course, to apply these concepts to movement disorders, models of movement disorders. So let's begin with a description of basal ganglia, a little bit more detailed than what Elena has done yesterday, but this was, of course, in our agreements. So the basal ganglia, as you all know, are subcortical nuclei. They are a number, of course, of nuclei tightly interconnected. We have a main input structure, which is the striatum. In the human, it's divided into the caudate and the putamen. And then you have two segments of the globus pallidus, an internal and an external part, the subthalamic nucleus, and of course, both substantia nigra pars compacta and pars reticulata. Of course, you will see that this loop is closed through the thalamus and back to the cortex. So, traditional views of basal ganglia function predict the existence of parallel loops, and mostly two major ways, loops generated by distinct medium spiny neurons. That is the major neuronal subtype of the striatum. So the medium spiny neuron may express D1 dopamine receptors or D2 dopamine receptors, and this gives rise to a so-called direct pathway and indirect pathway, respectively. The first one is thought to facilitate movement, and the other one is thought to inhibit movement. And as a consequence, the imbalance of these two is thought to generate movement disorders. Now, this is a very, let's say, it's neuroscience not for kids, but for scholars. And as you will see, it's much more complex. So if we just want to give a look at this scheme, but you have already seen some, and I will also shortly show you how more complex it looks like. So the cortex is connected to the striatum gives rise to the two pathways, as I said, the direct pathway to the GPI or through the GPE and STN. Then they go again to the substantia nigra. This connects, of course, these are inhibitory pathways that go to the thalamus and then the thalamus to the cortex. This is just a brief overview, and we will go through very soon through it. Anatomically, this is a monkey. Anatomically, you will see how they are located, just to have a rough idea, although Elena has shown some much more fancy image of this. And this is interesting, and this is something that I would like to remind, is that from an evolutionary point of view, this is, the basal ganglia are an area, a complex of regions that are absolutely highly preserved throughout the ontogenic development of the species. And this is of relevance. So, as we said, from the original, let's say, descriptions, there have been a number of experimental but also clinical evidence, for instance, those obtained by means of DBS, that have, let's say, posed some questions about the validity of these models. And so, after all, it's just more complex than what was previously guessed. And so if we want to go to the functional anatomy, I will show you how things started in the late 80s, mostly by Mellon, DeLong, but also by the group of Albin, Young, and Alexander and Krucher. These are just reviews, but in few years before, they originally described and proposed models of basal ganglia function. And in this original view, this is the long paper, as you see, there is a cortex with the supplementary motor area, premotor cortex and motor cortex that project to the putamen, and then you get the two direct and indirect pathways, and then back to the thalamus and to the cortex. So if you look at simple diagrams nowadays, they are not different from that. But something has changed because, first of all, there has been a discovery of the hyperdirect pathway from the cortex to the directly to the STN, and then many, not only the PPN, the pedunculopontine, but also other brainstem nuclei and the thalamic nuclei, let's say that they have been shown and then they've been demonstrated to play a major influence in the circuitry. So now we have something a little like this, even more complex than this, actually. If we look for the novelties, let's say, you can see that most of them arise in the brainstem, like the pedunculopontine nucleus, or in the thalamus with the centromedian and parafascicular nuclei. They project back to the striatum, and they have a very important role. One of them, just to give you a hint of this, it's been shown that, I mean, just to make you figure out what, so the centromedian and parafascicular nuclei are relay nuclei. So they receive sensory inputs. So if you are, for instance, if you are walking and you have to be careful when you cross the road, and there is a sudden acoustic signal, then these guys immediately start firing and send to the striatum an information that breaks or interrupts the normal corticostriatal transmission. So the motor task you are doing will be interrupted, and you will have to change motor program. So it's a very fundamental task. And you can see here a little, even more, let's say, detailed and so complex diagram. You see again here, look at PPN and brainstem, look at the different nuclei of the thalamus, so centromedian, parafascicular, ventrolateral, and relay thalamic nuclei, more in general, which means, I mean, you can easily guess that all these novelties came out because of the novel techniques, imaging techniques, but not only, also optogenetics, because before these were basically not accessible because there are many nuclei in a very small brain region. So it is, I mean, still it is very difficult to understand their basic function. But nevertheless, they have been now included in this scheme. So one of the elements that we have to take into consideration when we talk about basal ganglia is their parallel organization. So it is now clear that we have at least 3 different loops, which are anatomically, let's say, segregated. So you can see that the motor loop, you can see that starts from the motor cortex, then go to the putamen, GPI, and then again and back to the cortex. There is a limbic area and there is an associative cognitive area, which involves, let's say, different parts of the caudate and putamen. And this is something that fits very well with what Elena Moro has described yesterday, especially regarding the side effects of DBS. And this also a very nice reconstruction with the functional MRI, which shows you the different distribution of the caudate putamen loops. So this is the somatosensory motor. This is the limbic area, the yellow, and the associative area is in violet. So this implies at a functional level and also pathologically that we have an expanding universe of basal ganglia disorders. Because if we thought that just Parkinson's disease or Huntington's disease were disorders of the basal ganglia, now we are aware that OCD, Tourette syndrome, even some psychiatry formerly considered pure psychiatric conditions can be considered disorders of the basal ganglia. And this is based on these parallel loops that are always belong to the basal ganglia, actually. So this is, I think this is of extreme interest. Now let's go back to the let's say to the original work, to the original role of basal ganglia in movement. So how do they work? How do they facilitate or, let's say, program, how do they program movement? There are a number of different theories. One of these is the movement selection. So, according to this theory, one of the pathway, in this case the direct pathway, selects a movement. And the indirect pathway and probably also the hyperdirect pathways, they inhibit the, let's say, the surrounding patterns, movement patterns, to let the movement selection go straight. Other concepts also include the automatic running of motor programs, or even the formation and then the release of habits. So the movement is seen as actually an habit. This is conceptually a very thin line. I will probably, not probably, I will tell you more later on, but just to show you, for instance, in this piece in this scheme, that when you have a selection and then a chunking, then it becomes eventually an habit, one action that is repeated over time. And yes, this is the point where I wanted to get to probably give you a better idea. So in this very recent review article in Brain by Ikosaka, this is something that came out very in May. So I would suggest you to go through it. So first of all, they clearly indicate that there is a functional segregation in the caudate head and the tail. You see? Rostral and caudal. So they seem to have a different role in acquiring actions, or at least the memory for motor programs. So, and you can see here, the rostral basal ganglia together with the association cortex helps learning quick voluntary movements that eventually become then, let's say, subconscious, and in this case we can translate it into habits or automatic movements. Of course, we can switch from one to the other. I mean, if we are, let's say, doing a sort of multitask work, you are driving and you are you're answering your mobile phone, which happens, shouldn't happen, but in that case, you are performing two let's say, automatic actions. Those motor programs are running independently, differently from if you are cooking for the first time something and you're supposed to answer the phone. Just imagine that you cannot do either you burn your cooking or otherwise you answer something wrong to the phone, or you don't have the right attention to respond to the phone in an appropriate way. I think this happens commonly. So if you are busy, if your motor program is, let's say, is not automatically running, your brain will be, your striatum will be very busy. Otherwise, it will run an automatic program. Any question? This is rather intuitive, actually. Of course, it's not so intuitive to learn what are the mechanisms behind. Anyway, so how do these circuits work, actually? So one theory is that there is a center and surround mechanism. And the direct pathway is a net excitatory pathway, and the indirect pathway is a net inhibitory pathway. So the direct pathway can be a center, and the indirect pathway can be the surround. This is something that has been proposed a while ago, and you can see that here is the supposed selected motor pattern. It has to go through, but of course it has many obstacles. And how can it go through without any impediment? It's because the indirect pathway and the other, let's say the other components of the basal ganglia, they allow the motor program to go straight. So there is a movement selection and the center surround mechanism that allows the program that you choose to go directly. This is particularly true also when we will see you have a motor dysfunction. Because, for instance, in the case of tics, what we think, or at least what is thought nowadays, is that you have a disruption of this center surround because there are many ectopic centers, let's say, that starts simultaneously to fire, and so the selection is impaired, which fits with this hypothesis. So we talked about the direct and indirect pathways. This is something that you have learned in your textbooks, which of course is of great help in formulating these different hypotheses. However, you have to be very careful to this respect, also because very recently this is something that has been put forward by the group of Rui Costa formerly in Portugal, now in the States. They perform very elegant experiments with optogenetics in vivo. So just implanting a flashlight on the cortex of the animals and then just either activating or inhibiting the direct or the indirect pathway. And so they came to the conclusion that actually to have an action initiation, you need both pathways, which is a further complication, as you can imagine, because it goes beyond all the previous theories and all of what I said so far. And also, this is also on the same line of reasoning, the group of Paolo Calabresi as again, this is also an interesting review of the state of the art of this never-ending story. And I would focus on this. Intrastriatal connections are critical, and the two pathways are connected. So, do we have an experimental basis for this? Yes. Look at the recent papers by this group. This guy, Cazorla, number of papers. This is a neuron paper last year. So what they found is that, you see, striatal D2 receptors actually regulate the density of collaterals that bridge direct and indirect pathway. So, medium spiny neurons, you will see in a moment, they have a very wide dendritic tree. So they have collaterals, and they eventually bridge, that allow to bridge direct and indirect pathway. So this is an explanation to the Rui Costa finding regarding the need for both pathways. But at the same time, it tells you that things are even more complex than what we said so far. But this is very interesting and also has some implication for psychiatry, for schizophrenia. That if you, the concept is that in patients with psychiatric disorders in which you give D2 receptor antagonists for years and years, you interrupt this communication between direct and indirect pathway. And so it can cause you some side effects like the motor disorders you were mentioning yesterday. So, to make things even more sophisticated, of course, we have to think how these pathways communicate to each other through, of course, neurochemicals. And so we have, if we start from motor cortex, we see that motor cortex and the striatum, it's a glutamatergic pathway, excitatory glutamatergic pathway. Then the striatum, at least the projection neurons of the striatum are all GABAergic. So you have an inhibitory, a net inhibitory synapse to globus pallidus and substantia nigra reticulata. And these are again GABAergic that go back to the thalamus. And the last pathway back to the cortex is again glutamatergic. Of course, in all this context, you have to put dopamine, which is a crucial element and gives rise to the clinical symptoms when you take it off. And also, if we want to look at corticostriatal synapses, you can see how rich it is. You have, besides the dopaminergic projections, of course, you have a cholinergic intrinsic innervation, and then you have a number of cannabinoid receptors, adenosine receptors, serotonin receptors, besides calcium channels. So let's try to put all this into the main input station of the basal ganglia, which is represented by the striatum. So this is the striatum. You see the two glutamatergic inputs. One is from thalamus and the other is from cortex. And then you see the dopaminergic inputs from substantia nigra pars reticulata. And then you have the main neuronal subtypes. You have the two major spiny neurons. So one is the D1 expressing neurons, the other is D2 expressing neuron, and then you have three distinct GABAergic interneuron subtypes. One is expressing parvalbumin, the other is expressing carrotinin, and the other one is expressing neuropeptide Y and nitric oxide synthase. And then we have a a big cholinergic cell. So striatal architecture is quite complex. If we want to give a better look at striatal cholinergic interneurons, you see that they are, of course, receiving dopaminergic signaling. They are receiving cortical and thalamic glutamatergic inputs, and they can be modulated, their activity, by spiny neurons and fast spiking interneurons. So we have also to take into consideration the receptor distribution. So if you have the D1 GABAergic, the D1 GABAergic spiny neurons, they express D1, which is connected to G-OLF and cyclic AMP. However, also the D2 receptor, let's say the D2 receptor expressing spiny neurons, so the so-called indirect, the neurons that generate the indirect pathway, they express A2A. And A2A is also linked to G-OLF and cyclic AMP. So this chemical characterization can also, let's say, explain why the concurrent activation of the two pathways can be is taking place. Okay, this is again to say that the spiny neurons is receiving inputs from the local interneurons, but then from midbrain dopamine neurons and the glutamatergic fibers. And just to show you a very brief gallery of pictures that in our lab we took in the past few years. So we have a slice preparation, a striatal slice preparation. This is a coronal slice, and this you can see the spiny neurons filled with biocytin, and you can also record from parasagittal slice. It's a different angle that preserves the better preserves the corticostriatal connections. And you can see one of the dendritic trees studded with the spines. And this is the same at different enlargements. Again, this one, just to show you, just to tell you how dense is the dendritic tree of these neurons and how, so they most likely generate these collaterals that bridge the one with the two expressing spiny neurons. And also by means of stereotactic injections of viral vectors, you can also, for instance, GFP, you can also, color spiny neurons and go with a much higher resolution to look and see the spines, which express, of course, also the markers for glutamatergic afferents. Let's see if this works. Just to, don't know. This is a reconstruction of again, to give you the idea of how dense is the arborization of these cells. Okay. I mean, it was not so fancy, but still, at least gives an idea. As we mentioned, of course, this is not the main, this is not the only cellular subtype. It is the main projection neuron, the spiny neuron. But still you have a number of interneurons, as we mentioned. The cholinergic, so this account for about 5% of the striatal neuronal population, because 95 is made of spiny neurons. However, this 5% is crucial for a correct functioning of the striatum. And this 5% is made of either cholinergic cells, as we said, we will see even better later, cholinergic interneuron, and then the 3 types of GABAergic cells. Cholinergic interneurons are the, let's say, probably the most studied, also because from a technical point of view, it is still not easy to recognize and to pick all the single interneuron subtypes. Also because the amount of these cells is very, very low. And so cholinergic interneurons are cells that are autonomous pacemakers. It means that they fire spontaneously with a variable frequency between 3 and 9 Hz in the absence of movements. And they are peculiarly related to reward. It means that they stop firing when, and they anticipate the reward of a given, this has been shown in monkeys in vivo. So they are thought to correspond to the so-called TANs, so the tonically active neurons. These are, in fact, they fire spontaneously and then they stop when there is a reward that is coming. And they have, of course, some peculiar electrophysiological properties. I won't go into this detail because otherwise you definitely fall asleep. But just to show you again the chat staining in the striatum, this is, you see it's scattered throughout the striatum. And so you see they are kind of sparse, but still they are most effective. And here is another image with DAPI. You see that compared to the other neuronal subtype, they are, yes, they are scattered throughout the striatum. And we can see them by means of infrared video microscopy so that we can go with our pipettes and record them without big problems. And you see that they have this firing activity, tum, 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 which is rather peculiar for the striatum. No other cell is behaving like this when you record the cell in the striatum. And then they have, yes, these features like the seg. When you give a hyperpolarizing current pulse, it brings back the membrane, the current membrane, and it's a so-called seg conductance. Dopamine and acetylcholine interact in the striatum. And this is a crucial balance which is of relevance for movement disorders, we will see later on. And how do they behave? So if you give muscarine, which is, of course, a cholinergic muscarinic agonist, you see that these cells stop firing, hyperpolarizing, and then eventually, when you stop to perfuse muscarine, they go back and they start again to fire. So they have a self-inhibition through M2 and M4 autoreceptors. What happens if you give quinpirole? Like Emiliana was saying yesterday, D2 receptors, they modulate the activity of cholinergic interneurons by reducing their activity and so reducing also the release of acetylcholine and striatum. This is an essential requirement. Otherwise, acetylcholine would be released and produced excessively. So from this, this is not, maybe the light is a bit too much. Anyway, so here you have schematically spiny neurons, cholinergic interneurons, and then you have these rare GABAergic cells, interneurons, of course. If we want to give a quick look to this further interneuron subtypes, you see that fast spiking interneurons, they express parvalbumin, they have they can fire at very high frequency, more than 200 Hertz. And when you give a lower stimulus, they fire in a peculiar way, which is start firing, then stop, then they start again. In the case of these cells, LTS means low threshold spike. It means that the amount of current that you need to make this neuron fire is very low. And this starts very rapidly to fire. And then there is this type of cell that is very, very rare, very hard to find in the mosaic of striatal neurons. So, but functionally, what is the role of these interneurons? Because otherwise, this is a boring part of the story, which can be boring even to electrophysiologists. So, the functional meaning of this is that they all inhibit spiny neurons. If you give a quick look to these double recordings, these are double recordings, so you have here an FS neuron, interneuron, and here a spiny neuron. You see that if the FS fires, the spike of the spiny neurons is delayed. And again here, and again here. So there is basically a feed-forward and feedback inhibition. So GABAergic interneurons exert an inhibition on spiny neurons. And this is their role. So in summarizing the role of these cells, we have that fast spiking interneurons are involved in the control of spike timing. So it is taught that they regulate the timing in which the spiny neurons have to fire. Whereas the cholinergic interneurons are much more complex in terms of modulation, because they have, besides the dopamine acetylcholine balance, they also activate nicotinic receptors, which are expressed throughout the striatum. Of course, because acetylcholine activates also nicotinic receptors. So, going to plasticity. Emiliana, how? 10 minutes? Maybe I can introduce plasticity, and then we stop when at the last, the movement disorder. So, very briefly. I think not many of you, and since it is a school, I think it's very important to let you know who coined the term synapse. And this was the Lord Sherrington, Nobel Prize for Physiology, who said there is this description. In view, therefore, of the probable importance physiological of this mode of nexus between neuron and neuron, it is convenient to have a term for it. The term introduced has been synapse. These are his words. And so synapse because synapto in Greek means connect. Synapto. And so I think this is something that it's better for you to know in general. But why plasticity? Because plasticity is something that the term itself says that it's relevant for our brain to learn something. And so, and in fact, it is the case with motor learning, and which is the major pathway that is involved in this process of learning. Of course, the corticostriatal pathway. And because, as we said, it's involved in the formation of these procedural memories, like we said, we talked about memories or automatic movements, involuntary movements. These are all stored as files in a computer. And so we run the program, we run the motor program when we open that program. And this is something that has been shown even in humans. This is a recent paper in 2014. This group, by means of functional MRI, just teaching or saying to the people just to perform a task and seeing how they learn to perform this task, they see that the activity in the putamen, the functional connectivity, was strengthened progressively as participants learned the task. In the putamen, in the striatum, which is a confirmation of what has been shown in vitro. So we record exactly in this context, in this corticostriatal preparation, stimulating the cortex and recording in the striatum. If you do so, you get an excitatory postsynaptic potential, which is the expression of this of this synapse. And you can undergo either a long-term depression, so that the synapse will be tuned down, or otherwise, in the absence of external magnesium, which opens an MDA receptors, you get an LTP, a long-term potentiation. If you give, at this point, a train at low frequency, you are able to revert the synapse to a resting level, and this is called synaptic depotentiation. What is synaptic depotentiation? Just to give you a brief explanation. When you learn something or experience something that is redundant and doesn't need to be remembered, to be stored, you have to eliminate this type of memory. And eliminating a memory is an active process, is not a passive process. So depotentiation is what we need to bring back the synapse to a level that can be, in principle, then again used to go either LTD or for LTP. But then you have to be, let's say, to go back to a resting state. It's a sort of backup of what you do in your, for instance, when you sleep. Sleep is necessary because you do a backup of your daily activities. Some are removed, some are stored. And if some of these catch your attention, then it's possible that you wake up. If you remember something that has been through the day, and it's something that was important to remember, you can wake up at night and say, oh, there was something that I needed to do. Because your brain is backing up, is doing a sort of, is doing a depotentiation. So it's an active process. And this is also something very conserved in the ontogeny. It's interesting to see. This is something that it's just a notion, but it's very important. So we have something that stays there and works in the same way since, not since yesterday, some millions of years ago. And so, plasticity, as we said, it's important it's a paradigm for motor learning, but it's also relevant because it helps us defining some endophenotypes to specific movement disorders, either in, as we will see, in monogenic Parkinsonism, but also in dystonia. So our readout, of course, is the plasticity deficit, which can be very different from one movement disorder to another. And this is something that we will talk about later, I think. Yes. I stop here? Yeah. Okay. So after lunch is even worse. But let's try to keep the alertness. So we try to delineate which is the organization of the basal ganglia, And the neurochemistry and also what happens in the striatum with different cellular subtypes, and then plasticity. Of course, this was very general information because plasticity has many mechanisms, many complexities inherent to the fact that both pre- and postsynaptic elements are involved. So, of course, this is out of the scopes. I mean, I won't go into the details of this, but I wanted to then to turn into a functional readout, and especially by showing you the results we got in models of movement disorders. And in particular, as I said, there are a couple of models that we have been studying in the last 10 years, I would say. One is the monogenic Parkinsonism, so genetic forms of Parkinsonism, and the other one is one model of dystonia, of generalized dystonia. So also under this perspective, the previous speakers in some sense helped me. For instance, if you think of the generalized dystonia, yesterday Elena Amoro has shown some videos of young patients implanted with DBS. So now I will show you some of the data we got from a rodent model. So when we talk about basal ganglion and involuntary movements, this is a relatively old review paper by Jonathan Mink, but it's very impressive in its capability of foreseeing what could have been the results in the let's say in the 10 years that passed since he wrote. And again, he was proposing this idea of a competing motor patterns, which means that once you select an action to be performed, then the rest, let's say, of the circuit makes it happen, because it needs synaptically to deactivate or inhibit some of the areas of the surrounding areas. So what happens in the pathological conditions? So if we have, let's look at these circles. If you have a normal condition, let's say that this is the GPI, so one of the major, if not the major, output of the basal ganglia that then goes to the thalamus and back to cortex. So in this case, you have, you see, a normal motor pattern generator. What happens, for instance, in dystonia? In dystonia, you have, you see, this increase in the motor pattern generator. In chorea, in Huntington's disease, you have different ectopic foci, can we say in English, foci? Interesting. Which can generate motor patterns in an aberrant way. While in tics, you have, again, these aberrant generators, but these are stereotyped. So they are always the same. In the case of chorea, you instead have distinct patterns of motor patterns that occur, of course, aberrantly. And this is a nice model. So the traditional model of basal ganglia of Parkinson's disease, of animal models of Parkinson's disease, is the use of toxins. And most, especially in rodents, the 6-hydroxydopamine lesion in the medial forebrain bundle, which eliminates most of the dopaminergic innervation of the striatum. So what happens is that you lose TH staining in the substantia nigra, so you lose nigral neurons, and then you have a number of consequences. There is a severe motor impairment with the loss of bidirectional plasticity. So you lose both LTP and LTD. And then you give back levodopa to these patients, and in the short term, you have a recovery of motor function, recovery of plasticity, But in the long term, you can get dyskinesia. And so dyskinesia is also coupled to some, let's say, some aberrant plasticity. This is, and first of all, I wanted to recall one of our collaborative works a long time ago, actually, Emiliana. It was almost 20 years ago. And this was because Emiliana generated, she was the first to generate this D2 knockout model. And of course, we were extremely interested to see what was going on in the striatum. And in fact, plasticity was totally gone, accordingly to what she has shown yesterday. But then you have a couple of nice review. One is the group of Jim Surmeyer, and the other is always from Paolo Calabresi. And these are, as I said, a combination of information that may be I would say, complete if you are interested in going through this topic. However, besides the toxin models, as you certainly know, the elucidation of novel genetic components has totally changed the view and the way to approach Parkinson's disease at the research level in terms of experimental plans and experimental approach. And this has been, of course, pioneered by Maria Grazia, that has discovered what we have been discussing all the yesterday and today. And this, is, of course, has been one of the very first steps towards the amplification of this genetic Parkinson's disease, genetically determined. What is clear now is that although some, in the case of alpha-synuclein, is basically certain, I would say, but in the case of most of the other genes, we cannot say that it's the disease is caused by those genes. We can say that those genes are risk factors, which means not really much so far, but at least is a step forward to understanding some. And in fact, you can see here, LRK2 is a risk factor. And you see, they name it genetic factors, but still risk factors, with the exception, of course, of these synuclein models that are indeed causative of a monogenic Parkinsonism. And then you have a combination of environmental factors. You have heard from other speakers about epigenetics, and that is probably the key in reading. You know, epigenetics is like an orchestra director who reads the music, but the players is the DNA, and they play the same, but the director reads it differently. And this is a nice review by the Dawson and Dawson, and they describe all the models available so far, in this case the vertebrates, because we have invertebrate and vertebrate models. And so without going into the detail, here we have a number of autosomal dominant mouse models, starting from the alpha-synuclein models, and then we have recessive models, Parkin, DJ1, PINK1, and they describe also the electrophysiological and the neurochemical changes and biochemical changes that have been described. And then they also go through the viral models, which are certainly more novel as compared to the traditional rodents. And so we said we have invertebrate and vertebrate model. The invertebrate Drosophila and C. elegans, of course, they are relatively simple to study for a number of practical reasons. But nevertheless, the mouse has become the preferred system to study, and also especially because it allows to reconstruct a circuit. Although it's a rodent circuit, it's not a primate, but still it is informative. So there is a, maybe this will wake you up. So we have to realize what to expect from an animal model. And I think this notion is important for students, for trainees. Why? Because we will always, forever in your life, receive criticism about the fact that a mouse is not a man. And clinical symptoms, this is the critiques that clinicians move to us. And I am a neurologist, so I get the critics and I try to always to bypass this and to circumvent this criticism. Anyway, so these investigations, of course, and these models are not meant to reproduce the human disease in full. This is something that is may sound strange to you. But at the same time, you have to realize that you cannot expect to get the same, for instance, the same syndromes and the same clinical features that humans have, because these are different species. So, however, a useful animal model has to be judged by how effective, and of course, it can be replicated by others, can be used to explore pathophysiology and therefore identify targets for treatment. This is something that may be given for granted, but not always is like this. And this is, I think this kind of note of caution must be done all the time that you deal with animal models, especially rodents, of course. If you have a primate, maybe it's easier to sell the story. And you just think of when you study Parkinson's disease in Drosophila. So these are Nature science publications. Why? Because they are able to reproduce something that is absolutely compatible with what happens in humans. So for rodents, it's almost the same. So we, in the recent past, now it's quite a long time, so we collected evidence, and our readout, as I said, is an electrophysiological approach with plasticity experiments in the striatum. And we studied several models like DJ1, Parkin, and Pink1. More recently, we focused on Pink1. So specifically on it's a rare disease, it's a rare condition, much more rare than Parkin itself, because Parkin is the most frequent among the recessive forms. But we started working on the Pink1 deficient mice, so the knockout, the full knockout mouse. And we found an impairment of plasticity, an impairment of dopamine release, which was kind of expected. Although there was no symptom, no nigral degeneration, no, let's say, no overt symptom. But then we turn to something that, to us, is much more interesting, and it's the heterozygous condition. So these papers, these were relatively recent, but at the same time, they were pioneering the idea to look at heterozygous non-manifesting carriers. So the patient who has the mutation, of course, is a clinically manifesting patient. But there are a number of carriers who do not manifest the disease, and eventually they will develop some clinical symptoms. But at the same time, to understand and to go and look for a disease-modifying therapy, you have to look at this condition, in a pre-manifesting, preclinical condition. And so this, for instance, if you look, this was another paper, probably the first one in 2002, that were analyzing this with fluorodopa PET patients and non-manifesting carriers. And after 12 years, they observed that most of the dopamine of the non-manifesting carriers, they now present a mild Parkinsonian phenotype. So they were exactly the persons to study and to recruit for a clinical trial for a disease-modifying therapy, a really disease-modifying therapy. So this has been also a topic for a nice review in which they try to understand which is the role of this heterozygous mutation. And you can see that for these genes, this is the frequency of Parkin, PINK, and LRK2. But most interestingly, this is the penetrance. You see? So look at homozygous LRK2 mutations, 100% penetrant. It means that if you have the mutation, for sure you will develop Parkinson's disease. But then, if you already start looking at heterozygous, always LRK2, between 13 and 80, and if you look at PINK, between 1 and 25, what determines this penetrance? We don't know, and I think this is the trick, the key. What makes these guys go to the clinical manifestation and or otherwise which preserves them from being clinically manifest? We also know that, you see, this is a DAT scan, so it's the uptake of dopamine from nigrostriatal terminals in normal conditions, and then you see a long disease progression. You see how it more and more loses the ability of because the terminals are dead. And so you see how it goes down along the years, along disease progression, the amount of dopamine in the striatum, which means, first of all, that dopamine, the level of dopamine is clinically relevant. So the symptoms has a direct correlation with the amount of dopamine present in the striatum. So the question was, can we monitor disease progression with a model of heterozygous mice? And we did this type of observation in heterozygous PINK1 mice. So in these mice, we observed that nigral cell count and also physiology was normal. This is a TH staining, as you can see, was no significant change. And you can see also that dopaminergic neurons in the substantia nigra, these are actively firing. They don't have different responses to dopamine. This is a classical response, an autoreceptor response, which blocks their firing activity. So they are inhibited by dopamine. And nothing seems to change. Also, the behavior, locomotor activity was normal in this PINK1 heterozygous mice. Then we went to study striatal physiology, striatal neuron physiology, and basic properties. You see there. Basic properties. You see firing activity, and then spontaneous activity was pretty normal in the two strains. But then, when we went to give a high-frequency stimulation to induce LTP or LTD, we saw that while LTD was normal, we saw that LTP had a subtle deficit. It was less evident, less expressed compared to normals, you see. So it was not fully expressed. So what does this mean? It means that, we measure dopamine level, and we found that it is already decreased. So which means that the terminal is already somehow impaired. And this is absolutely in line with those fluorodopa PET studies in patients. Then, we tried to rescue this with the levodopa or SKF, and especially with amphetamine. You see, amphetamine, of course, is a dopamine releaser. So you just boost dopamine release and you rescue the plasticity deficit. And this was done with tyramine and selegiline and rasagiline. So without going into the detail, selegiline and rasagiline are MAO inhibitors, MAO-B inhibitors. So they block the degradation of endogenous dopamine, while tyramine actually is one of these previously known as false transmitters, now better known as traceamine. Tyramine is the most relevant of traceamines, and if you give traceamine and if you give tyramine, it behaves like amphetamine, and so it rescues this subtle plasticity deficit. And also, they are able to restore the levels of dopamine, and this was expected. So, which is the link between the two? So, you have, as I said before, the previous speakers helped me in this task because they already explained and identified how relevant is ATP and energy production for a release process. So what happens, we think that ATP production may represent the link between mitochondria, and PINK1 is as a mitochondrial localization, and dopamine release. And this is quite, let's say, obvious. So we think that we have a sort of a lazy synapse. We called it lazy synapse just to figure out what is what goes on, and this is very roughly what previously has been shown by Chandra. And in this case, you have a sort of a blockage of release just because you don't have enough energy to sustain dopamine release. And if you force it with amphetamine, of course, you get it. So, of course, then we don't know how this condition may be preceding the nigrostriatal terminal death. But certainly, it's important to know that already in the heterozygous condition, you have a functional deficit. This is important, at least I guess this is important, and also important to remind how we should consider an animal model, the relevance, even if there is no phenotype. So this first part, okay, is concluded, and just a few minutes to give you an idea of dystonia, of what happens in dystonia. So, you know, you have seen some of these pics yesterday. So we go from simple, let's say, simple focal dystonias like writer's cramp, you see, or musician dystonia, for instance, to generalized forms, which are, unfortunately, most of them are genetically inherited, but most of them occur in early adolescence. So it's a very dramatic condition. And this, in particular, the DYT1, is the most frequent. Of course, it's a rare condition, but among the 25, it's DYT1 to 25. So you can imagine how many genetic forms we have now. And this form is inherited as an autosomal dominant, so it's a critical condition, but with a low penetrance again. These cases are induced by, as you have heard yesterday, by a 3 base pair deletion resulting in a mutation of torsin A, basically. It's important to know that we have no evidence for neurodegeneration. So it's a completely different from Parkinson's disease or from Huntington's disease. And this is also the reason why when you put a stimulating electrode, when you implant these guys with the DBS and you do DBS in these guys, this is absolutely the only way to prevent them from having this life. So before DBS, these guys were dying for several complications like respiratory failure or internal complications, and they used to die very early in their life. So there are a number of these animal models. Of course, we tried to focus on the YT1, and what we found, we started working on cholinergic interneurons. Then I will tell you why. We started recording these cells and trying to see which was the relationship between acetylcholine and dopamine. This was what I was mentioning this morning. You remember that quinpirole in normal condition is causing a decrease in the firing activity of these cholinergic cells and reducing local acetylcholine release. So we have 3 models now, maybe 4. Yeah, 4 actually. 4 models now in which we observe the same alteration. There is an increase in firing activity upon quinpirole application. So there is an aberrant paradoxical excitation, which means that dopamine is not doing what it should, that is, reducing the activity of acetylcholine neurons in the striatum. And this was interestingly coupled to a reduced level of D2 and a reduced activity of, in the GTP gamma SA, you see the activity of G proteins was reduced in the striatum of these mice. So we have a functional impairment, which consists in this aberrant excitation, coupled, a bit strange, but anyway, coupled with a decreased expression. And also, the functional evidence for their alteration was also provided by this model, the catalepsy, the haloperidol-induced catalepsy. So haloperidol normally induces a catalepsy, and this is a model to test the efficacy of D2 receptors, the activity of D2 receptors. And you can see that in these mice it was dramatically reduced. So, and again, we then, from this model, these were transgenic mice, you see, it's a transgenic model of overexpressing mutant torsin A, and then we shifted to knock-in mice, a more, let's say, physiological model, more reliable model. And again, we observe a significant decrease of D2 receptors and also RGS9. RGS9 is immediately following the signaling machinery of D2 receptors. So the signaling, the pathway, the signaling pathway, the dopaminergic pathway is completely gone in these mice. So to go quickly, which is the impact of this aberrant cholinergic change on plasticity in the striatum? First of all, why? Because here in patients and in non-manifesting carriers as well, several indications say that there is an impaired sequence learning of motor performance, and cortical plasticity is again different in patients. So it looks like we are on the right track in this sense. And in fact, what we see, in normal condition, you give high frequency stimulation in the absence of magnesium, you get LTP. Then you get depotentiation, or otherwise you can get LTD. So this is a normal bidirectional plasticity. What we observe in DYT1 knock-in mice is a huge LTP. without any sealing. So there is a loss of homeostatic plasticity. This plasticity is incredibly increased. There is no break. In fact, you cannot depotentiate anymore. You give a low frequency stimulation to depotentiate the synapse, no way. And you never observe LTD. So the main conclusion is just that we have a loss of inhibition, which is exactly what is described in the clinical neurophysiological papers, when they describe in patients a loss of inhibition and an increased plasticity. It is exactly what happens in our mice. So going backward, why is acetylcholine important in this case? Because besides DBS and surgery and the chemodenervation, we don't have much in our armamentarium in terms of medical treatment of this patient. But we have anticholinergics. So why do we have anticholinergic? We had some evidence, clinical evidence, old clinical evidence that these drugs were effective in dystonia. But why? Which is the rationale? The rationale is that we have, as we have been saying yesterday, today, we have a balance in the striatum between dopamine and acetylcholine. And if you disrupt this balance, then symptoms come out. So, and this is something that has been observed long ago. This was pioneering studies by Ornikiewicz and by others. So, and in fact, as you have seen from the posters that have been yesterday, have been shown, yes, Milian.
Why is it should be more specific of these cholinergic neurons?
No, torsin is widespread. Torsin is an AAAA ATPase, and it makes a lot of things. So we still don't know what it does. But as I've shown, we made the paradoxical excitation that we observed in this knock-in, we observed it also in cholinergic specific knockouts for torsin A. So that deletion is cell specific. That deletion, mutation of torsin A in the cholinergic cell gives rise to that behavior, electrophysiological behavior. So just to, very briefly, just to say that anticholinergic, either very selective ones or some not, let's say, M1 preferring antagonists, were able to rescue all the deficits that we had. So both LTD, not LTP, but synaptic depotentiation. So through which mechanism? So to reconstruct all the scenario, what we think is that normally dopamine, you see, we have dopamine in the striatum. When dopamine goes to D2 receptors and blocks acetylcholine release. And so you have a physiological plasticity. In dystonia, what we get is an aberrant excitation by dopamine, which causes a further increase of acetylcholine that excites M1 muscarinic receptors on the dendritic tree of medium spiny neurons. And this is the idea we finalized to explain this dopamine and acetylcholine deficits. So yes, basically this is a further summary with M1 expressed on the spines of the dendrites of medium spiny neurons, and this excessive acetylcholine. So we believe that there is an hypercholinergic state in dystonia. And as you can see, talking about the reliability of models, this is completely different from the pink one. nothing to deal. And the mechanism, so if I give an anticholinergic to pink 1, nothing happens. So the mechanisms are completely different and as expected. Okay, so in conclusion, yeah, as I underlined in a previous part, there is much complexity that requires probably further revision of the model of basal ganglia function, especially in disease state. I think that to this respect, we still have to understand which is the real role played by interneurons and probably also by glial cell, astrocytes, which have an active role, for instance, in buffering transmitters. And so, however, in spite of these limitations, we think that these models help advancing our knowledge of the basic mechanisms underlying these disorders. And then, okay, this is just to thank all the people of the lab, and this is our awful university, and this is also the foundations where most of our labs are. Thank you.